Class 11, Chapter 10, Cell Cycle and Cell Division, the most important chapter and one of the basic chapter. Are you aware that all organisms, even the largest, start their life from a single cell? You may wonder how a single cell then goes on to form such large organism. Growth and reproduction are characteristic of cells, indeed of all living organisms. All cells reproduce by dividing into two, with each parental cell giving rise to two daughter cells each time they divide. These newly formed daughter cells can themselves grow and divide, giving rise to a new cell population that is formed by the growth and division of a single parental cell and its progeny. In other words, such cycle of growth and division allow a single cell to form a structure consisting of millions of cells. Cell cycle. Cell division is a very important process in all living organisms. During the division of a cell, DNA replication and cell growth also takes place. All these processes, that is cell division, DNA replication and cell growth, hence have to take place in a coordinated way to ensure correct division and formation of progeny cells containing intact genomes. The sequence of events by which a cell duplicates its genome, synthesizes the other constituent of the cell and eventually divides into two daughter cells is termed cell cycle. Although cell growth in terms of cytoplasmic increase is a continuous process, DNA synthesis occurs DNA synthesis occurs only during one specific stage in the cell cycle. The replicated chromosomes DNA are then distributed to daughter nuclei by a complex series of events during cell division. These events are themselves under genetic control. Phases of cell cycle. A typical eukaryotic cell cycle is illustrated by human cells in culture. These cells divide once in approximately every 24 hours. However, this duration of cell cycle can vary from organism to organism and also from cell type to cell type. Yeast, for example, can progress through the cell cycle in only about 19 minutes. The cell cycle is divided into two basic phases, interphase and M phase, that is mitosis phase. The M phase represents the phase when the actual cell division or mitosis occurs, and the interphase represents the phase between two successive M phases. It is significant to note that in 24 hours average duration of the cell cycle of a human cell, cell division proper lasts for only about an hour. The interphase lasts more than 95% of the duration of the cell cycle. The M phase starts with the nuclear division corresponding to the separation of daughter chromosomes, karyokinesis, and usually ends with the division of cytoplasm, that is cytokinesis. The interphase, though called the resting phase, is the time during which the cell is preparing for division by undergoing both cell growth and DNA replication in an orderly manner. The interphase is divided into three further phases: G1 phase, S phase, and G2 phase. G1 phase corresponds to the interval between the mitosis and initiation of DNA replication. During G1 phase, the cell is metabolically active and continuously grows but does not replicate its DNA. S or synthesis phase marks the period during which the DNA synthesis or replication takes place. During this time, the amount of DNA per cell doubles. If the initial amount of DNA is denoted as 2C, then it increases to 4C. However, there is no increase in the chromosome number. If the cell had diploid or 2n number of chromosomes at G1, even after S phase, the number of chromosomes remains the same, that is 2n. In animal cells, during the S phase, DNA replication begins in the nucleus and centrioles duplicates in the cytoplasm. During the G2 phase, proteins are synthesized in preparation for the mitosis while the cell growth continues. Some cells in the adult animals do not appear to exhibit division. Example, heart cells. And many other cells divide only occasionally as need to replace cells that have been lost because of injury or cell death. These cells that do not divide further exit G1 phase to enter an inactive stage that is called quiescent stage, that is G0 stage of the cell cycle. Cells in this stage remains metabolically active but no longer proliferate unless called on to do so depending on the requirement of the organism. In animal, mitotic cell division is the only seen in the diploid somatic cells. However, there are few exceptions to this where haploid cell divide by mitosis. For example, male honeybees. Against this, the plant can show mitotic division in both haploid and diploid cell. From your recollection example, alteration of generation in plants, identify plant species and stage at which mitosis is seen in haploid cells. M phase. This is the most dramatic period of the cell cycle, involving a major reorganization of virtually all components of the cell. Since the number of the chromosomes in the parent and progeny cells is the same, it is also called as the equational division. Though for convenience, mitosis has been divided into 4 stages of nuclear division: karyokinesis. It is very essential to understand that cell division is a progressive process and very clear-cut lines cannot be drawn between various stages. Karyokinesis involves following 4 stages: prophase, metaphase, anaphase and telophase. Prophase, which is the first stage of karyokinesis of mitosis, follows the S1, S and G2 phase of the interphase. In the S and G2 phase, the new DNA molecules formed are not distinct but intertwined. Prophase is marked by the initiation of condensation of chromosomal material. The chromosomal material becomes untangled during the process of chromatin condensation. The centrosome, which had undergone duplication during S phase of the interphase, now begins to move towards the opposite pole of the cell. The completion of prophase can thus be marked by the following characteristic events. Chromosomal material condenses to form compact mitotic chromosomes. Chromosomes are seen to be composed of 2 chromatids attached together at the centromere. Centrosome, which has undergone duplication during interphase, begins to move towards the opposite pole of the cell. Each centrosome radiates out microtubules called esters. These two esters together with the spindle fibers forms mitotic apparatus. Cells at the end of prophase, when viewed under the microscope, do not show Golgi complexes, endoplasmic reticulum, nucleolus and the nuclear envelope. And now metaphase. The complete disintegration of the nuclear envelope marks the start of the second phase of the mitosis. Hence, the chromosomes are spread through the cytoplasm of the cell. By this stage, condensation of chromosomes is completed and they can be observed clearly under the microscope. This then is the stage at which morphology of chromosomes is most easily studied. At this stage, metaphase chromosomes is made up of 2 sister chromatids which are held together by the centromere. Small disc-shaped structures at the surface of the centromeres are called kinetochores. These structures serve as the sites of the attachment of spindle fibers formed by the spindle fibers to the chromosomes that are moved into position at the center of the cell. Hence, the metaphase is characterized by all the chromosomes coming to lie at the equator, with 1 chromatid of each chromosome connected by its kinetochore to spindle fibers from one pole and its sister chromatid connected by its kinetochore to a spindle from the opposite pole. The plane of the alignment of the chromosomes at the metaphase is referred to as the metaphase plate. The key feature of metaphase are spindle fibers attached to the kinetochore of the chromosomes, and the second, chromosomes are moved to the spindle equator and get aligned at the metaphase plate through spindle fibers to both poles. Now, anaphase. At the onset of anaphase, each chromosome arranged at the metaphase plate is split simultaneously, and the 2 daughter chromatids, now referred to as the daughter chromosomes of the future daughter nuclei, begins their migration towards the 2 opposite poles. As each chromosome moves away from the equatorial plate, the centromere of each chromosome remains directed towards the pole and hence at the leading edge, with the arms of the chromosomes trailing behind. Thus, anaphase stage is characterized by the following key events. Centromeres splits and chromatids separate. Chromatids move to opposite pole. Telophase. At the beginning of the final stage of karyokinesis, that is telophase, the chromosomes that have reached their respective poles decondense and lose their individuality. The individual chromosomes can no longer be seen, and each set of chromatin material tends to collect at each of the two poles. This is the stage which show the following key events. That is, first, chromosomes cluster at opposite spindle poles and their identity is lost as discrete events. Nuclear envelope develops around the chromosomes cluster at each pole, forming two daughter nuclei, nucleus, Golgi complex, and ER reform. Cytokinesis. Mitosis accomplishes not only the segregation of duplicated chromosomes into daughter nuclei, karyokinesis, but the cell itself is divided into two daughter cells by the separation of cytoplasm called cytokinesis, at the end of which cell division gets completed. In an animal cell, this is achieved by the appearance of a furrow in the plasma membrane. The furrow gradually deepens and ultimately joins the center, dividing the cell's cytoplasm into two. Plant cells, however, are enclosed by a relatively inextensible cell wall. Therefore, they undergo cytokinesis is by a different mechanism. In plant cell, wall formation starts in the center of the cell and grows outward to meet the existing lateral walls, and the formation of new cell wall begins with the formation of a simple precursor called the cell plate that represents the middle lamella between the walls of the adjacent cells. At the time of cytoplasmic division, organelles like mitochondria and plastids get distributed between the two daughter cells. In some organisms, karyokinesis is not followed by the cytokinesis, as a result of which multinucleate condition arises, leading to the formation of syncytium, liquid endosperm in coconut. Now, significance of mitosis. Mitosis or the equational division is usually restricted to the diploid cells only. However, in some lower plants and in some social insect, haploid cells also divide by mitosis. It is very essential to understand the significance of this division in the life of an organism. Are you aware of the some examples where you have studied about haploid and diploid insect? Mitosis usually results in the production of diploid cells with identical genetic complement. The growth of multicellular organism is due to mitosis. Cell growth results in disturbing the ratio between the nucleus and the cytoplasm. It therefore becomes essential for the cell to divide to restore the nucleocytoplasmic ratio. A very significant contribution of mitosis is cell repair. The cells of the upper layer of the epidermis, cells of the lining of the gut, and the blood cells are being constantly replaced. Mitotic division in the meristematic tissue, the apical and lateral cambium, results in the continuous growth of the plants throughout their life. Now, second one, meiosis. The production of offspring by sexual reproduction, including the fusion of two gametes, each with a complete haploid set of chromosomes. Gametes are formed from specialized diploid cells. This specialized kind of cells division that reduce the chromosome number by half result in the production of haploid daughter cells. This kind of division is called meiosis. Meiosis ensures the production of haploid phase in the life cycle of the sexually reproducing organism, whereas fertilization restores the diploid phase. We come across meiosis during gametogenesis in plants and animals. This leads to the formation of haploid gametes. The key features of meiosis are as follows. First, meiosis involves two sequential cycles of nuclear cell division called meiosis 1 and meiosis 2, but only a single cycle of DNA replication. Meiosis 1 is initiated after the parental chromosomes have replicated to produce identical sister chromatids at the S phase. Meiosis involves pairing of homologous chromosome and recombination between non-sister chromatids of homologous chromosomes. 4 haploid cells are formed at the end of meiosis 2. Meiotic events can be grouped under the following: prophase 1, metaphase 1, anaphase 1, and telophase 1, and in meiosis 2, prophase 2, metaphase 2, anaphase 2, and telophase 2. Now, meiosis 1. Meiosis 1, prophase 1. Prophase of the first meiotic division is typically longer and more complex when compared to the prophase of the mitosis. It has been further subdivided into following 5 phases based on the chromosomal behavior, that is leptotin, zygotin, pectin, diplotin and dikinesis. During leptotin stage, chromosomes become gradually visible under the light microscope. The compaction of the chromosomes continues throughout leptotin. This is followed by the second stage prophase 1 called zygotin. During this stage, chromosomes start pairing together and this process of association is called synapsis. Such paired chromosomes are called homologous chromosomes, and electron micrographs of this stage indicates that chromosome synapsis is accompanied by the formation of complex structures called synaptonymal complex. The complex formed by a pair of synapsed homologous chromosomes is called bivalent or tetrad. However, these are more clearly visible at the next stage. The first two stages of the prophase 1 are relatively short-lived compared to the next stage, that is pectin. During this stage, the 4 chromatids of each bivalent chromosomes become distinct and clearly appear as tetrad. This stage is characterized by the appearance of recombination nodules. The sites at which crossing over occurs between non-sister chromatids of the homologous chromosomes. Crossing over is the exchange of genetic material between two homologous chromosomes. Crossing over is also an enzyme-mediated process, and the enzyme involved is called the recombinase. Crossing over leads to the recombination of genetic material on the two chromosomes. Recombination between homologous chromosomes is completed by the end of pectin having the chromosomes linked at the sites of the crossing over. The beginning of the diplotin is recognized by the dissolution of the synaptonymal complex and tendency of the recombined homologous chromosomes of the bivalents of the separate from each other except at the site of crossovers. These accept structures called chiasmata. In oocytes of the some vertebrates, diplotin can be last for the months or years. The final stage of the meiotic prophase 1 is dikinesis. This is marked by the termination of chiasmata. During this phase of chromosomes are fully condensed and meiotic spindle is assembled to prepare the homologous chromosomes for separation. By the end of dikinesis, the nucleus disappears and the nuclear envelope also breaks down. Dikinesis represents transition to metaphase. Metaphase 1. The bivalent chromosomes aligned on the equatorial plate and the microtubules from the opposite pole of the spindle attached to the kinetochore of homologous chromosomes. Anaphase 1. The homologous chromosomes separate while sister chromatids remains associated at their centromeres. Telophase 1. The nuclear envelope and nucleolus reappear. Cytokinesis follows. This is called as dyad of cells. Although in many cases of chromosomes do undergo some dispersion, they do not reach the extremely extended state of interphase nucleus. The stage between the two meiotic divisions is called interkinesis and is generally short-lived. There is no replication of DNA during interkinesis. Interkinesis is followed by prophase 2 as much simpler prophase than prophase 1. Now meiosis 2. Prophase 2. Meiosis 2 is initiated immediately after cytokinesis, usually before the chromosomes have fully elongated. In contrast to meiosis 1, meiosis 2 resembles a normal mitosis. This nuclear membrane disappears by the end. The nuclear membrane disappears by the end of prophase 2. The chromosomes again become compact. Metaphase 2. At this stage, the chromosomes align at the equator and microtubules from the opposite pole of the spindle get attached to the kinetochore of the sister chromatid. Anaphase 2. It begins with the simultaneously splitting of the centromere of each chromosome, which was holding the sister chromatid together, allowing them to move towards the opposite pole of the cell by shortening of microtubules attached to the kinetochore. Now telophase 2. Meiosis ends with the telophase 2, in which the two groups of chromosomes once again get enclosed by a nuclear envelope. Cytokinesis follows, resulting in the formation of tetrad of cells, that is 4 haploid daughter cells. Now significance of meiosis. Meiosis is the mechanism by which conservation of a specific chromosome number of each species is achieved across the generation in sexually reproducing organism. Even though the process paradoxically results in the reduction of chromosomes number by half, it also increases the genetic variability in the population of organisms from one generation to next generation. Variation are very important for the process of evolution. So that's it. Thank you so much.